I'm going to talk to you guys about DNA replication. That's the process of making, or excuse me, process of DNA making a copy of itself. Before we begin this, I want to start with a bit of vocabulary for you. There's quite a few enzymes that are involved in this process, and I want to make sure you've got that first. So let's go. There we go. All right. So DNA replication. So as you can see here, we've got a nucleotide here. We have our base and our sugar and our phosphate, and you can see that they're actually numbered 1, 2, 3, 4, 5. And the reason why I've ' this here is so that you can see that we read DNA from the 3' end to the 5' end. So if there were several nucleotides in a row, we would start at this end and we would read in that direction, reading the other nucleotides. So if we read from 3' to 5', we build in the opposite direction. We would actually build a 5' to 3' molecule next to it. So then that'll make more sense as we go. So let me give you an idea of what we're talking about here. So if I've got a double-stranded DNA molecule and I separate those two strands, and I've got a molecule here. This one's 5', and this is 3'. So we're talking about these two strands. And at the other end of the 3' is the 5' end. And on the other end of this one is the 3' end. So we're going to read this from 3' to 5'. Same thing we're going to do here. We're going to read this from 3' to 5'. So reading from 3' to 5' here is not a problem. We just go continually from one end to the other. And we create 5' here to 3' here. Now, that works out really well, but we have to read this other strand from 3 to 5'. And then when it opens up again, we'll read again from 3' to 5'. And then when it opens up again, we'll read again from 3' to 5'. So here we've got our new DNA strand with 3' at this end and 5' at this end. So we end up with these fragments, and these are called Okazaki fragments. And we'll talk about how those fragments actually get joined, because we can't leave this gap and this gap here. We've got to actually join those together, and we'll talk about how that's done in the next video. But let's move on with enzymes. So there are several enzymes that are involved in this process. The first one is topoisomerase, and you can see that's here. That's this circular molecule. What happens is it encircles the DNA and it will actually relax the supercoils. It doesn't uncoil the DNA itself, the double helix, but it does relax the supercoils in the DNA. The next molecule, excuse me, as you can see here, is DNA helicase. And what DNA helicase does is it works like a wedge between these two strands. What it will do is it will push between these two strands. It will do two things. One, it uncoils the double helix, and then it will also break the hydrogen bonds between those two strands, separating them out. There are also a couple of molecules here, these circles. These are called single-stranded DNA binding proteins, abbreviated SSB. So these actually attach to the DNA along the outside of each of the strands, and what they do is they keep these two strands from coming back together, because that's the natural attraction that they will have for each other. So you can see here we've got we have two nucleotides. We've got one up here and we've got one down here, and what we're going to demonstrate here is an enzyme called polymerase. Polymerase is actually a category of enzymes that synthesize long chains of polymers together, and the polymer we're talking about making right now is a nucleic acid. So that's why we've got a nucleotide up here and a nucleotide down here. The specific polymerase that we're going to work with is DNA polymerase. Let me see if I can get myself out of the way here. Is DNA polymerase. DNA polymerase will actually covalently bond this nucleotide here with this nucleotide here, forming this nucleotide strand, or this strand, which will eventually be a DNA strand. It covalently bonds the two nucleotides together with what's called a phosphodiester bond. That's a bond that's very specific to DNA. And we'll talk about how that happens. So again, talking about the 3' and the 5', you can see we've got the 3' in our sugar, and we've got the 5' carbon in our, coming between our sugar and our phosphate here. So we're going to bind a 5' to a 3' here. So again, we read in the 3' to 5', but we build 5' to 3'. So we're actually building, see if I can do this, build in this direction. So when we do this, when we combine this nucleotide with this nucleotide, this is called a condensation reaction. That should sound familiar. You should recognize that. A condensation reaction, as we talked, what we talked about a couple of slides ago, you should recognize it from the beginning of the semester when we were talking about proteins. We also then talked about condensation reactions. Condensation reactions happen when we have an OH here and we have an OH here. Sorry about that. We take one of these OHs and the H from the other side. They come out and they form a molecule of water, and we're left with the other oxygen that then bonds this phosphate group to this deoxyribose sugar. And this creates what's called a phosphodiester bond, because there's two ester bonds in this molecule. So it's called a phosphodiester bond, but you need to know it's a covalent bond. The other thing the DNA polymerase does is it also proofreads the DNA. So it covalently bonds and it proofreads the DNA. So there are two other enzymes that you need to know about. One is ligase, and this molecule, this yellow molecule right here, and this yellow molecule right here, these are both ligase molecules. Ligase molecules actually join Okazaki fragments. Those are the fragments that we drew earlier. And remember I said we can't have these two fragments detached from each other. We've got to have one smooth strand of DNA, just like we have this one smooth strand of DNA right up here at the top. We need to be able to join this red segment with this red segment and this red segment and this growing red segment here. This is the polymerase that is actually building or adding these nucleotides here, and this is the polymerase that's building in that direction. And you can see that this is building in this direction, but it's building in segments. So it built this one first, then it built this one second, then it built this one third and fourth, and then what it's eventually going to do is it's going to build one here. And what we need to do is we need to attach those together. That's the responsibility of ligase. Ligase joins these fragments together, and those fragments are called Okazaki fragments. And again, this is another dehydration synthesis or dehydration reaction or a condensation reaction. So every time we bond this strand with this strand or any of these strands together, we have a byproduct of water. There's one other molecule that I want to talk to you about. It's not an enzyme, and that's primer DNA. Primer DNA is very important in DNA synthesis, and without it, you can't begin to build the DNA. So RNA, excuse me, primer RNA is an RNA that initiates DNA synthesis. The reason why you can't synthesize DNA without it is because DNA polymerase will bind the 3' and the 5' ends together. Well, it can't do that if there is no 3' end. So the primer RNA actually provides that 3' end, and that's what these molecules right here are. This pink in here, this pink in here, and this green here, and this green here. Those are the primer RNA. So the primer RNA, again, are these locations here, and the ligase is bonded around it. And what the ligase does is it removes the primer, fills in the nucleotides, binding that together, joining the Okazaki fragments together. Since the helicase has unwound to this point, what would happen is once this polymerase is done here, it's going to jump to this area, and it's going to again create a new Okazaki fragment going that direction. And that's it. What we're going to do is we're going to go through and we're going to draw this. So if you would get out a blank sheet of paper before you start the next video, you're going to need a blank sheet of paper and probably the lined sheet of paper that you were just using. And I'll see you.